Okay, so it's that time again. Here is my e-report for section 2. So the topics of discussion are going to be the cell membrane structure and the different classes of proteins that are found within the cell membrane, how proteins are transported to the ER and into the nucleus. We will discuss the formation, docking, and fusion of Catherine-coated vesicles, cell crawling, cell junctions, and we'll finish up with the psychopathiness of cancer. So first up is the cell membrane structure. So the cell membrane is made up out of a lipid bilayer, lipid meaning fat and bilayer meaning two. So there's two opposing lipid layers found in the cell membrane, which is shown to the picture to the right. So the lipid bilayer is a barrier between the inside and the outside of the cell, and it works by blocking the diffusion of most hydrophobic molecules. So the hydrophobic regions of the phospholipids are the tail regions, which are found in the middle of the membrane, and those are the water-insoluble regions. The hydrophilic head regions of the phospholipids are water-loving regions, which are seen by the purple circles. The hydrophobic properties of the tail regions of the phospholipids is what drives their need to stay together. So because they repel water, they want to be far away from water. So by sticking all of their hydrophilic tails together, the phospholipids can force the water out because they have the hydrophilic heads to work as the barrier to keep the water out. So the same thing works for the head regions of the phospholipids, and this gives them their amphithetic properties so that they always have this tendency to cluster together to repel the things they don't like. So the cell membrane is a 2D fluid, which allows for the movement of phospholipids laterally side to side. So fluidity is important in a cell membrane because the cell is constantly moving. The particles are being absorbed and excreted, and the cell itself needs to be able to move. The fluidity of the membrane depends on its composition, so it depends on how closely or loosely the phospholipids are packed together. So the image to the left shows how the packedness of the phospholipids can affect its fluidity. So the more viscous the cell membrane, the more closely packed the phospholipids are. On the other side, the more fluid the membrane, the less tightly packed the phospholipids are. And this is due mainly to the number of double bonds that are found in the tail regions of the phospholipids. The double bonds cause the phospholipids to kink out, as shown in the picture. And so when they're kinked like that, they can't pack as closely together. So that leaves room in the membrane, which makes the membrane be more fluid. This allows it to adapt to different cellular environments that the cell might be placed in. Cholesterol is often used to fill in these gaps that are created. So the cholesterol, in this case, would take the place of those gaps and would stiffen the membrane up so that it's less fluid. So there are many other components that make up the cell membrane. These also include proteins, which are shown in this picture. There are several different kinds of proteins that span the cell membrane that are only partially integrated into the cell membrane. So we're going to go over those now. Membrane proteins play a big role in the activity and function of the cell membrane. More than 50% of the cell membrane is made up of proteins, and their main function is transport. So their job is to move molecules across the membrane that wouldn't be able to cross the membrane. These also include channel proteins and transport proteins. Proteins can be anchoring proteins or receptors. So they can be found in different parts of the cell membrane, and like phospholipids, proteins can also have hydrophobic regions and hydrophilic regions. There are three main types of proteins found in the cell membrane, and those are transmembrane proteins, integral proteins, and peripheral proteins. Transmembrane proteins are proteins that extend through the whole cell membrane. They have hydrophilic regions on each end, which is how they're stuck in the membrane and stay there. Integral proteins are proteins that are embedded into the membrane that associate with a hydrophobic part of the membrane. So they can be partially embedded or fully embedded like transmembrane proteins. Peripheral proteins are proteins that are not embedded into the cell membrane at all. They are either covalently linked to a phospholipid head on the cell membrane, or they are non-covalently associated with another embedded protein. So this can all be seen in the picture to the right, where integral proteins are labeled. So there's one that spans the whole membrane and one that's only partially embedded. And it also shows you peripheral proteins that in this case is attached to another protein. Carbohydrates are also found on the cell membrane. Carbohydrates are sugars that are found in different regions of the cell membrane. So glycoproteins are proteins that are found with sugars attached to them. Seen in the picture below, over to the left on the blue, it shows a carbohydrate attached to a protein, which is a glycoprotein. Glycolipids are basically the same thing, except it's a sugar attached to a lipid. So those are shown to the right. The little green structure shows a lipid attached to a carbohydrate. These protect against environmental damage for the cell membrane, and they also absorb water. So they also aid in cellular recognition and play a big role in cell-to-cell adhesion and communication between cells. Next, we're going to talk about protein transport into the endoplasmic reticulum, or the ER. The ER is the entry point for proteins going to other organelles. So proteins that are traveling to the Golgi or the lysosome, the cell surface for secretion, or to the ER itself. So from the ER, proteins are ferried in transport vesicles to their end destination. Proteins enter the ER before translation is complete, which is why there's two sections of the ER. There's the rough ER and the smooth ER, and this is why the rough ER is embedded with ribosomes. The interior portion of the ER is the lumen, which is where the hydrophilic, free-roaming proteins are entered into the ER. Translocation of the protein begins at a free ribosome in the cytosol. So when an ER signal sequence, which is found at the amino terminal end of a new protein, emerges, the amino acid chain is brought to the cytosolic surface of the ER with the ribosome attached. So this can be seen in the picture. All the ribosomes that are in the ER are there because that ER signal sequence emerged during translation. A signal recognition particle, or SRP, is present in the cytosol that recognizes and binds to the ER signal sequence found on the amino acid chain. The SRP interacts with an SRP receptor that's on the surface of the ER membrane. This slows down the translating ribosome and allows for the SRP to find and interact with the ER-bound receptor. The SRP is then released and the amino acid chain is threaded through a translocation channel into the ER. The ER signal sequence on the protein opens the translocation channel. The ER signal sequence remains bound to the channel the entire time, which means the protein is translocated into a looping fashion through the channel. During this process, the signal sequence is chopped off the protein, which allows the protein to be completely threaded into the ER lumen, and then the signal sequence is degraded. So this whole process is shown in this image. So we see up to the left top, we see the RNA being translated into the protein, and we see the signal sequence that signaled the attachment of the SRP, and so the SRP was brought down and met with the SRP receptor so that the amino acid chain can then be threaded through the translocation channel protein. So that is seen in step 3. And step 4 shows how the ribosome keeps translating the amino acid chain and how it's threaded loopwise into the lumen of the endoplasmic reticulum. Eventually, not shown, but in between step 4 and step 5 is when the red signal sequence would be chopped off and degraded so that the rest of the protein that was created can be released and flowed into the endoplasmic reticulum. Transmembrane protein into the ER begins basically the same way. However, somewhere in the middle of the protein is a stop transfer sequence. So when this region gets threaded through the channel, the region is hydrophobic. So because it's hydrophobic, it gets stuck when it enters the channel. When this happens, it gets released laterally into the ER membrane, and the signal sequence is released and degraded. Another thing that could happen is when the ER signal sequence is embedded eternally into the amino acid chain. So this is referred to as a start transfer sequence. So this is never removed or degraded, and there can be multiple start or stop transfer sequences found in an amino acid chain. So when this happens, there would be many regions threaded through the membrane instead of just one. So this is shown in this picture. So in this little black box is what would happen if there was just one stop transfer sequence found in the protein. So we see that the SRP has bound to the SRP receptor and that the SRP was attached to the signal sequence. And so we also see that the amino acid was transferred through the channel, and that as the amino acid was being threaded through the channel, a stop transfer sequence was found, and because of that, the hydrophobic region of the protein was just pushed laterally out into the membrane of the ER, which, as you can see, has happened right here. However, just because the protein was pushed into the membrane, the ribosome doesn't stop translating the amino acid chain. So translation still occurs, and the excess protein is just threaded out on the other side of the membrane. So the bottom of the picture shows what would happen if there were multiple start sequences in the amino acid chain. So when the start transfer sequence would reach the translocation channel, the protein would be looped to the other side of the membrane because that signal sequence would be stopped in the channel, and the amino acid would be threaded through. So that's what the bottom picture shows, and it would have multiple parts of the protein embedded into the membrane with excess pieces on either side. Protein transport into the nucleus is a very different process. Nuclear proteins can only enter through nuclear pores. This is because the nucleus is surrounded by its own membrane, which is a double membrane. This membrane is the nuclear envelope that is made up of two phospholipid bilayers. Nuclear pores allow for the only access into and out of the nucleus. Nuclear pores are small complexes that are filled with water, and they allow only small hydrophilic molecules to pass through. Large molecules need nuclear localization sequences, which are recognized by nuclear transport receptors. Nuclear transport receptors recognize the sequence and bind to it. The receptors interact with the pore fibrils and allow passage of the protein through the pore. And when it's done, they are released and the receptors are cycled back. So in this picture, you can see what we just discussed. You can see the protein that is trying to get through the channel, and you can also see the nuclear localization, which is the red dot that is attached to the protein. So as the protein reaches the pore of the nucleus, the nuclear transport receptor, it binds to the red dot, which is the localization signal, and helps that protein get transferred through the pore. And as you see it going through the pore. And at the end, when the protein is into the nucleus, we see that the nuclear transport receptor is released and is cycled back through the pore to repeat the process over and over. Next, we're going to talk about vesicle transport. Vesicle transport is very, very important to the cell. Transport vesicles carry soluble proteins by a process of budding and fusion to other organelles and membranes throughout the cell. There are different pathways that transport could be done in, such as the secretory pathway, which is when things are made in the ER and sent outwards into the cell, or the endocytic pathway, which is when things are brought into the cell from the outside. Vesicle formation begins with a clathrin protein coat. Vesicles normally bud off from the Golgi for secretion, or they can bud off from a cell membrane for endocytosis. The first thing that happens is that a pit forms that is covered with clathrin. Then, as the vesicle forms deeper, a protein called dynamin forms a ring around the neck of the vesicle and constricts until the vesicle pinches off from the membrane and becomes its own unit. Adaptins are proteins that are used for cargo selection. They secure the clathrin cage to the vesicle membrane. Proteins have transport signals that bind to cargo receptors that are vesicle membrane-bound proteins. Adaptins capture specific cargo molecules by trapping receptors below the neck of the vesicle membrane. There are different adaptins for different membranes. So this picture shows the formation of a vesicle. So we see the cargo receptors and the cargo molecules, and this picture shows the light green adaptins that hold the cargo receptors in place. The dark green proteins is the clathrin coat that forms. And so right here we see that little pit that is forming. And so in the next stage we see the more circular shape of the vesicle with the dynamin in yellow that's constricting the vesicle until it's pinched off completely. And then we see the last stage, which is the vesicle that still has that clathrin coat. It still has the adaptins holding the receptors, the cargo receptors, and the cargo. And then that is the vesicle that travels along until it gets to where it needs to go. This picture goes a step further and it shows the uncoating of the clathrin coat with the adaptins, and this doesn't happen until fusion with its target membrane occurs. So we're going to talk about that in a little bit. The next step in vesicle transport is vesicle docking. So once the vesicle is made and has reached its target organelle, it must recognize the membrane and dock with it. Rab proteins, which are shown in the picture as yellow, are specialized proteins on vesicle membranes that serve as the membrane identifier. They are specific for different kinds of membranes and are recognized by tethering proteins, which are shown in green. So the tethering proteins interact with the Rab proteins and kind of bring the vesicle closer to the membrane that it's going to be docking with. Snare proteins, shown in blue and red, on both membranes interact and stabilize the docking proteins. So this picture shows T snares, which are the snares found on the target membrane, and V snares, which are shown on as red on the vesicle membrane. So you can see how these come together in the kind of fusion stage, and they start to twist and intertwine to pull the membrane closer together. The last and final piece of vesicle transport is fusion with the membrane. Vesicle fusion connects the vesicle to the target membrane and releases the vesicle contents into the membrane, or the cell that it's attaching to. The vesicle membrane becomes part of the target membrane, but before the fusion can occur, the clathrin coat is removed. Fusion can also be delayed. Only when a right cellular signal is given will fusion occur. So if this happens, the vesicle will be left in the docking position, and this is really important for secretory proteins because we don't want proteins or signals being released, like adrenaline, for example, when there's no reason for it. So this video describes the whole fusion process. So we see the tethering proteins connecting and the snare proteins connecting. The signal occurs to allow fusion to happen, which is that little yellow spark. And then we see the membranes connecting and fusing together. The vesicle membrane becomes part of the target membrane, and the contents of the vesicle is released into the cell. Our next topic is cell crawling, which is very important because cells need to have the ability to move throughout the body, like white blood cells, for example. They need to be able to travel to different parts of the body when they're signaled to, which affects healing. Cell crawling is due to cells sensing their environmental signals and migrating towards them using actin. Cell crawling is done in 3 steps, which is protrusion, anchoring, and advancing. The integration of signals from the environment affects a protein called Rho protein. Rho is a G protein that can trigger the actin polymerization and bundling for Flipopedia to form. It can also trigger lamellipedia formation, and it can trigger actin-myosin interactions. I apologize already for how I'm probably pronouncing these things. Actin-myosin contraction is powered by the attachment, release, and hydrolysis of ATP. So each of those processes results in the movement of myosin to actin, which is how the cell is able to crawl. So protrusion is driven by new actin polymerization. A thin sheet-like lamellipodia protrusions are pushed out, which contain a dense meshwork of actin. These are oriented so that filaments have the positive ends close to the plasma membrane. So this is seen in the picture that was shown before. You can see the difference between the laminopodium, which is the branched cross-linked actin filaments, and the filopodium, which are just smaller bundles of parallel filaments. So filopodia are thinner protrusions that are pushed out from the cell membrane, and they contain a loose bundle of actin filaments. They are oriented in the same way. Lamellipodia and filopodia are sent out all over the cell surface until they sense a chemoattractant and are able to anchor on a crawlable surface. Integrins are what bridge actin to the surface and allow for the anchoring process to happen. Advancement is made by the contractile forces that are made between actin and myosin, which is what allows the cell to pull itself along the cell surface. So this is seen in a video that I have.
The shape of the cell is highly polarized. A broad, thin veil of cytoplasm called the lamellipod is at the front of the cell, and the back of the cell is tapered into a thin tail. The nucleus and major organelles are located in the cell body. A network of actin filaments fills the entire cell, while contractile myosin molecules are located in the cell body. Cell crawling takes place in 4 stages. In the extension stage, the cell pushes the lamellipod forward. This movement is driven by rapid polymerization of actin filaments at the leading edge of the cell membrane. In the next stage, the lamellipod adheres to the underlying surface using specialized membrane adhesion sites. Firmly anchored, the cell is now ready to generate the force to move the cell body forward. The third stage is translocation of the cell body. This is accomplished by contraction of actin and myosin bundles at the lamellipod cell body border. After the cell body translocates, the back of the cell releases from adhesion sites and moves forward.
So that video does a good illustration of the whole process of cell crawling. Cellular junctions is our next topic. Cellular junctions are needed because they allow epithelial cells to connect to different things. Why is this needed? Because epithelial cells create sheets and layers of cells to form linings and barriers for our body and membranes and a whole bunch of things. Epithelial cells were compared to our body's cell membrane because it acts as a barrier. There are 5 types of cell junctions that we are going to talk about. There are tight junctions, hemidesmosomes, adherent junctions, desmosomes, and gap junctions. The picture to the right shows an example of epithelial cells that are sitting on top of connective tissue, and so in between each of those epithelial cells are the different junctions, which we are going to go over. And so this is a pretty good illustration because it shows the epithelium, it shows the connective tissue, and it shows how the cell junctions are used to hold together those epithelial cells so that they form a nice tight layer. So tight junctions are used to seal cells together so that virtually nothing can leak through. They are formed by 2 proteins, claudins and occludins, and they are arranged on both sides of the cell and form a zipper so that the cell can be joined. The proteins then zip up and seal the 2 cells together. Without tight junctions, life wouldn't be possible because molecules would leak through cells instead of traveling through them. Tight junctions are what are used to define membrane domain. So this picture shows a close-up of what one of the tight junctions look like. And so it literally does look like a zipper with the interlocking junctional proteins, which create an impermeable arrangement for the 2 cells. Hemodesmosomes, desmosomes, and adherent junctions, all 3 provide a sheet of epithelial cells with mechanical strength by joining individual cytoskeletons together. Adherent junctions and desmosomes bind one epithelial cell to another. They use the protein cadherin, which interacts tightly with the 2 cells, linking them together. Cadherins are tethered to actin filaments on adherens, or keratin on desmosomes, which links them directly to the cytoskeleton. Hemodesmosomes bind epithelial cells to the basal lamina. Integrins in hemidesmosomes are linked directly to collagen. So this picture is a good example of all 3 different junctions. So the big picture in the middle shows, again, the epithelial cell, and it shows how the junctions hold together adjoining cells. So to the left is the close-up of the hemidesmosome junction, and so the purple there is the basal membrane, and you can see how the hemidesmosome uses integrins to connect the epithelial cell to the basal membrane. To the top right, you can see the close-up of the adherin junction, which uses the cadherin protein, which connects to the actin filaments in the cells to connect them together. And underneath that are desmosomes, which use cadherin, and the cadherin is what connects to the keratin filaments that forms that close, tight barrier, which connects the cells. Gap junctions are the last junction we're going to talk about, and they are private communication systems that exist between neighboring cells. Gap junctions are actually openings. They are made through connexons that are transmembrane protein complexes that meet between two cells and create a continuous channel across the two cell membranes. Small inorganic and hydrophilic molecules can freely diffuse between cells. Cells are metabolically, chemically, and electrically coupled. Gap junctions are what creates the wave of muscle contractions across the entire heart muscle with each beat. So this picture is again a close-up of a gap junction, and you can see the channels that are formed in the intercellular space between the two epithelial cells. So our last and final topic is going to be about cancer and how cancer cells are like the psychopaths of society. Normal cells function like people in society. Most of them work towards a common goal. They listen to and obey environmental cues and signals to divide, so it's kind of like our laws. They live in large multicellular societies of constant molecule and information transfer, and they have no free will. So it's kind of like us, we wake up, we go to work, get money, pay our bills, and do everything over again. Cancer, though, is a complete violation of this social order. So cancer cells are selfish and asocial. They don't contribute to the overall whole of the organism. They only look out for themselves and their own self-survival. They crowd, push, and destroy tissues that are in their way, and they divide without requiring or needing any signals from the body. Cancer cells are relentless expanding clones of abnormal cells. Cancer functions in several ways. So cancer cells are mainly dangerous because they no longer require signals from other cells to survive. They also don't default to apoptosis. So in normal cells, if something happens, they sacrifice themselves for the greater good and they die. Cancer cells don't do that. They are mutated so that if something's wrong, they just keep dividing and they don't default to apoptosis. Cancer cells are also immortal. They don't have an age and they don't die. Whereas regular cells, they have an average lifespan and then they die and they get regenerated. Cancer cells are also abnormally invasive and they adhere to, survive at, and divide in foreign tissues of other cell types. So normally when they are put into other foreign areas, they can't survive at all because they don't have any of the same signals, they don't have any of the same environment to get nutrients. So if liver cell was placed into a lung cell environment, then it would just die because it wouldn't know what to do. But cancer cells aren't like that. They adapt and don't need cellular cues, so they can just do whatever they want and grow wherever they want. So that's what makes them very dangerous, and that's what gives them their psychopathiness, their ability to live and do whatever they want without any care for the greater organism is what makes cancer so dangerous. So that's it. That's the end of this report. Thanks for an awesome semester, and see you in genetics.